Hello students, in the last lecture we have been discussing regarding the plasma membrane structure. We have finished with the introduction of the plasma membrane. Next we have seen the structures associated with the plasma membrane along with the evolution of the structure of the plasma membrane, different models suggested by different scientists that we have discussed in the previous lectures. And today we will be discussing exclusively with the membrane components. One are the lipids and the proteins. If you remember, I have outlined the membrane structure in the last lecture, and in this lecture I will be discussing in detail with the membrane components. Okay, so first we will start with the lipids today. Once we will go through the structures associated with the plasma membrane in brief. So look at the structure, children. Here I have discussed with the lipid molecules. So this is the lipid bilayer, and along with the lipid bilayer we have many proteins which are arranged in the mosaic. Essentially we are talking about the fluid mosaic model given by Singer and Nicholson. According to them, these proteins are arranged in a mosaic pattern. They are irregular, interrupting the lipid bilayer. And in this we have discussed with the lipid molecules which are amphipathic in nature. They have both hydrophobic as well as the hydrophilic nature. Hydrophobic region is a tail region which is mainly made up of hydrocarbon, and it is highly non-polar in nature. Then what about the head region? Head region, it is hydrophilic in nature. Hydrophilic means it is water-loving substance, it is polar, and it is generally made up of phosphate. I have told you the alcohol and the phosphates are present. The lipids which are mainly seen in the membrane are the phospholipids. Okay, so these points we have discussed regarding the lipid molecules, and along with that we have discussed with the structures of the proteins. We have two type of proteins. What are the two type of proteins, children? Intrinsic and extrinsic proteins. Intrinsic means which are integrated with the membrane. In that again we have two types. Integral proteins are of two types. What are the two types of proteins? They can be partially embedded or they are superficially attached to the membrane. If they are superficially attached, they will be present like this, and they are known as the peripheral or ectoproteins, and they are partially embedded, which are known as the integral proteins. So integral proteins are very difficult to isolate, right? And the peripheral proteins, they will be attached with a weak bonding and they can easily be separated from the plasma membrane, right? And here in this integral proteins, again I have told you some are partially embedded. Okay, they are drilled into the membrane partially, and some are completely embedded into the membrane, drilling completely, forming channels and tunnels inside the membrane. And we have some other proteins which are having their domains which are exposed exterior and also in the interior region. So this is cell exterior as mentioned before, and this is cell interior towards cytoplasmic phase. And both of these phases, these proteins will be showing the domains. Protein domains will be present. These are the part of intrinsic proteins or integral proteins, and these proteins are also said to be the tunneling proteins or transmembrane proteins. Okay, so this is what we have discussed with the proteins in the membrane. And next we have discussed with the carbohydrate. Carbohydrates are exclusively present on the outer leaflet of the plasma membrane, and in this carbohydrates, again there are of two types. These carbohydrates are attached directly to the lipids, and those are known as glycolipid. Glyco is sugar, carbohydrate, and associated with the lipids, they together constitute glycolipids, and the process of this attachment is known as glycosidation. Okay, and next we have discussed with the proteins which are attached to the carbohydrates, sugar and protein binding together, they are said to be the glycoproteins. Okay, so carbohydrates, they will be present as they are individually. Carbohydrates are rarely seen here. Why? Because they are generally attached to the membrane. So on the membrane surface, what molecules will be present? The lipids. So they are generally present with attached with the lipids, or they may be present attached with the proteins. Okay, one is glycosylation, where the carbohydrate molecule associated with the protein, and one other is the glycosidation, which is the lipid molecules associated with the sugar molecules. Okay, So this is what we have discussed in the last lecture. Along with that, we have another important molecule which is giving the stability to the membrane, or you can say it will maintain the stability of the membrane, which is the cholesterol. Okay, it's a complex lipid. It is also a kind of lipid molecule, but those are the complex lipids. Okay, and these cholesterol molecules are present here and there, attached, and they are present in between these lipid molecules. Okay, so this is the thing we have discussed in the last lecture, and today we'll be discussing with the detailing of the lipid molecules. And in order to study this membrane, so many membranes have been studied by different scientists. In those days, they have used simple techniques like X-ray diffraction technique, and also they have studied this under the electron microscope. Okay, and nowadays we have different techniques to study this plasma membrane, and one of the best technique is the freeze fracture technique. So what is this freeze fracture technique? As mentioned earlier, this membrane, it is very thin. It is ultra thin in nature. It is very delicate, so that we cannot isolate this membrane, right? So let us say you will be having the cells with you, and now you want to examine the membrane, plasma membrane. Generally, the most explored membrane for this, in order to study the structure of the plasma membrane, is the erythrocyte membrane. Why? Because erythrocyte membrane, that is the RBC cells, they are very simple in its structure. Why? Because they are enucleated cells. We don't have any nucleus inside the cell, and hence they are easy to study. Okay, less complicated structure. And here in this structure, if you say these are certain cells, you have certain cells with you, and now the cells will be frozen at very low temperatures. So in order to freeze these cells, they will use a chemical substance which is known as liquid nitrogen, which have the temperature around -172 degrees centigrade. And at this temperature, these cells will get frozen. Once the cells they get frozen, they will be crystallized and it will become hard, right? Then what you will do, they will simply crack the membrane using a microtome knife. Okay, so let us say, so this is a cell and this got frozen using liquid nitrogen, and later they will fracture the membrane. First what they will do, they will freeze the membrane, and later what they will do, they will fracture the membrane. In its normal state, we cannot fracture this membrane, but when it is crystallized, when it is solidified, we can fracture this membrane. So how is this technique employed? This freeze fracture technique, the two terms which are given here you have to understand. One is freezing. Freeze means freezing the membrane, and later they will fracture the membrane. Okay, so what will happen when you freeze a membrane or freeze a cell? The cell will get crystallized. Okay, so once the cell is get crystallized, it will be very hard, right? Then what you will do, you will fracture the membrane using a knife, a microtome knife. And it is not important to learn this technique in detail, just for your understanding. I'm just explaining how these membranes will be isolated and studied. They cannot be isolated intact. So what they will do, they will crystallize the cells, and later they will break the cells so that the membrane, the lipid bilayer will get opened. Then they can easily study this membrane under a microscope, electron microscope. Okay, so this is how the membranes are studied. So what will happen, children? You will freeze this cell at very low temperature, and there is a chemical substance which is known as liquid nitrogen having -172 degrees centigrade of temperature will be maintained, and using this chemical the cells will be frozen and later they will be fractured at the membrane region. Once they have fractured the lipid bilayer, it will open like this, right? So let us say this layer, it is open. Once you have fractured the membrane, the layer will be opened, the bilayer, and you can see the structure which is like. So this is how you can see the membrane. So this is one of the best technique. We have different type of techniques which are used, but this is the one of the best technique which is used to isolate or study the membranes. So this is regarding the freeze fracture technique. Okay, now let us see how these lipid molecules will appear in electron microscope. What is their structure, what is their nature, what type of chemical groups which are present in this lipid molecules, that we will study. According to Singer and Nicholson, the membrane, it is not static. And what is the nature of the membrane? It is very oily and greasy. Why? Because it is having the fluid nature, as they are made up of the fat substance. Fats are always oil or grease like substances, and they are showing continuous movement that we have discussed in the last lecture. So all these membranes, all these lipid molecules and the protein molecules, they will be continuously moving in the membrane, and they will show the fluidity. They are highly viscous in nature. Even the Singer and Nicholson, they have studied the viscosity of the membrane. These lipid molecules, they were having the viscosity. Viscosity means the ability of the molecule to restrict its flow is known as the viscosity. So the viscosity experiments were mainly conducted by Singer and Nicholson in order to study the plasma membrane. So according to Singer and Nicholson, when they have done the viscosity experiments on this plasma membrane, they have observed that these lipid molecules, they are showing this viscosity. Viscosity means, I have told you, the tendency to restrict its flow. So what is the nature of the membrane? It is fluid in nature rather than the oil. So oil, we all know oil will be showing the flowing, it will be having the flowing ability, but these lipid molecules, they are like fluids. They are not like oil. Okay, so this was the hypothesis which was given by the Singer and Nicholson. Now let us look into the detailed structure of the lipid molecules. So we are discussing with the first component, that are the lipid molecules, and next we will be discussing with the proteins, enzymes and carbohydrates. So first we will take up with the lipid molecules. So look at the structure, children. So this is, let us say, this is the lipid molecule which is present in the membrane. And here we have two regions for the lipid molecule. We have the head region, the globular one, and we have the tail region, which is the linear one. Okay, so let us mark it. This is head and this is tail region. And generally, what is the chemical nature of the head region? Head region, it is highly polar. It can react with water, it can bond with water, it can create the hydrogen bonds with the water, and it is said to be polar in nature. Okay, head region, it is said to be polar in nature. As it is reacting with water, as it is having the ability to react with water, we call this region as hydrophilic region. So this region above and this region below, the head region, they will be in direct contact with the water. So this is cell exterior and this is cell interior. They are in direct contact with water, right? And hence we call this region as hydrophilic region. Okay, this is polar and hydrophilic region. Okay, and next we have the tail region. Generally the phospholipids it will contain the two tail regions. Yes, we do have only one fatty acid molecule. There you can see only single tail, but generally they have the structure. Okay, so the tail region, it is, what is the nature of the tail region? Non-polar and hydrophobic in nature. They will not be reacting with water. They are water-hating substance. What is a water-hating substance? I have told you, they are generally made up of hydrocarbon, hydrogen and carbon. It is continuously present and it is non-reactive with the water. Clear? So now, so what? Lipid molecule is nothing but a fat substance. Okay, it's a fat. Then what does lipid molecule or fat molecule it is made up of? That we will see. So I have told you that this particular tail region, it is mainly made up of hydrocarbon. Okay, so it is made up of hydrocarbon. And what about the head region, children? In head region, as the majority of the lipids are the phospholipids, I am writing it as phosphate. Okay, so where is the phosphate? Yes, we have the phosphate here. And here, remember, any type of substance can be present. That we will see later what type of substances can be present. X represents any type of substance. And generally the phosphate, it is attached to the main alcoholic region, that is glycerol. Okay. So look at this. This is a general structure of the phospholipid molecule which I have given. Now I will be discussing with the type of lipids present here. So we will have the tail region. We have two tail regions, and in this region it is non-polar and hydrophobic in nature due to the presence of the long chains of the hydrocarbon. And next we have the polar head region. This is head region, and in this head region in this head region you can see the presence of glycerol, which is an alcohol, and next we have the phosphate. Along with that we have some additional groups that are present that we will see later. So these three groups are present, you have to remember. Okay, so here we have three components and here we have two components. So this is a general structure of the lipids. And here in this lipid molecule, the outer surface, it is the polar, and the inner surface is the non-polar region. So if you compare with the membrane structure, the outer layer, the head molecules are oriented towards the outer surface, towards the cell exterior, and also in the lower leaflet of the plasma membrane, they are facing towards the cytoplasmic phase. Here even we have the water, the cytoplasm, and also we have the external cell environment is also made up of water. So both these polar regions are facing outside. Then what about the hydrophobic region, the tail region? They will be facing inside, they will be forming a core, the hydrophobic core, it will be made inside the plasma membrane. So this is how the lipid molecules are oriented. Now let us see what type of lipid molecules are seen in the case of the plasma membrane. So lipids, there are two major categories of lipids present on the plasma membrane. One are said to be the phospholipids and the second one are said to be the glycolipids. What are the first one, children? Phospholipids. Why? Because the lipids are associated with the phosphate like this, and such type of lipids are said to be the phospholipids. Okay, and phospholipids are the major class of lipids present on the plasma membrane. Other than that, we have the glycolipids, which are associated with the carbohydrate molecules. If the lipid molecules are associated with the carbohydrate, we call them as the glycolipids. So let us see the structures of these both categories of the lipids. Okay, so in order to understand the structure of the lipids, we have to generalize its structure. Then only it will be easy for you to study. So what is the generalization that I have given here? The lipid molecules, if you are talking about the lipid molecules present on the plasma membrane, they will definitely fall into this category. So this formula, it is mainly meant for the lipid molecules on the plasma membrane. So what components are present on this lipid molecules in the plasma membrane? They have mainly three components. What are the three components, children? One, they have the fatty acid molecules. It may be one tail or two tails like this. If there are two, we have the two fatty acid tails, or sometimes we have only one fatty acid tail. That will constitute the tail region. And this head region, it will contain three major groups. What are the three major groups? One, they have the alcohols, and in this alcohols again, the main alcoholic group will be present as well as subsidiary alcohol, means two alcoholic groups will be present in general. Okay, so the first one, it is said to be the the major alcohol, and the second one is said to be the minor alcohol or subsidiary alcohol. Along with this alcohol entities, we have a special group attached to this head region. And what is a special group? It can be any molecule like phosphate, or it can be a nitrogen, a sulfur, or a carbohydrate. Different type of molecules will be attached in the head region. Okay, so based upon that, we have the classification of lipids present on the membrane. If you compare the structure to the general structure I have given, I will explain this. So this is the fatty acid tail molecule. So this is tail region, and the head region, it contains three components. What are the three components? So this is, look into the structure, this is the glycerol. Glycerol is, let us say, a main alcohol of this lipid molecule. Okay, and along with the glycerol we have another alcoholic molecule that is present here. I have represented it as X. Okay, X means anything that I have given, but here let us say this is an alcoholic group. First major alcohol and this is the minor alcohol. And what is the third group I have told you? It's a special group. It can be anything like nitrogen, sulfur, phosphate, or the carbohydrates. Mainly the carbohydrates are present generally, and these are attached like this. Okay, so these three groups, they will constitute the head region, and the fatty acid molecules, they will constitute the tail region. So because of these groups, this is mainly polar in nature, and the fatty acids, they will be non-polar in nature. Okay, so now let us see the classification of lipids based upon the presence of the special group as well as the alcoholic groups. I have given the general structure of the lipid molecule here, and regarding the components present in the lipid molecules, the membrane lipids, I have given here the three components: fatty acids, alcohols, the major alcohol and the minor alcohol. Rarely this minor alcohol will be absent in certain type of lipids, children. So these are the two type of alcohols I have given, and the third component I have told you, the special group which is present will be attached with any one of the component. It can be phosphate, sulfur, nitrogen or carbohydrates. All of these molecules can be present as the extra group or the special group. Okay, so this is regarding the components of the membrane lipids, and this is the general structure of the lipid molecule. Here I have given the special group. S meant for the special group, and we have two alcohols, the major and the minor, or the substituent alcohol will be present, and we have the two fatty acid chains. And here I have not written, children, fatty acid, this will include the tail region, and these two components together constitute the head region. This is tail and this is the head region. So let us see what are the type of lipids that are present on the plasma membrane quickly. So first we have the first group of lipids I have told you, which are the phospholipids, and the second group of lipids, they are said to be the glycolipids. Due to the presence of phosphate, these lipids are said to be the phospholipids. So based upon which group, children? The special group, the extra group which is present, based upon that nature, the lipids are two types. Phospholipids if they have phosphate, glycolipids if the lipids are associated with the carbohydrate molecule. So look at these lipids. So these are the simple lipids. They are not at all attached to any other compound here. But there are certain lipid molecules which are attached to the carbohydrates. Yes, we have discussed in the previous video. I have told you the carbohydrate molecules are attached to the polar surface of the lipids. And these lipids are said to be the glycolipids, right? So these are the modified lipids. Lipids which are attached to the carbohydrates, they are said to be the glycolipids. So this is one category of the lipids present on the membrane. So this is regarding glycolipids, and coming to the phospholipids. Phospholipids in turn, they are divided into two classes. The first one being glycerophospholipids and the second one being sphingolipids. So what is the base for this classification here? Here, based upon the alcohol moiety, the major alcohol moiety, these lipids are classified into glycerophospholipids and sphingolipids. Listen carefully, children. So first I have told you, based upon the special molecule which is associated with the lipids are classified into two categories. What are the two categories? Phospholipids, and the other one is the glycolipids. Phosphate if it is attached to phospholipids, and glycolipids if carbohydrate it is associated with the lipid molecules, we call them as glycolipids, right? And again, based upon the alcoholic group, the major alcoholic group present in these lipids, they are again classified or subclassified into two categories: glycerophospholipids or sphingolipids. Then it is very easy to remember. Glycerophospholipids are the ones where you have the major alcohol component which is known as glycerol. Okay, glycerol is the alcohol, and this alcohol, it is present as a major component here. If you look at the structure of the glycerophospholipids, the majority of the lipids are of this type. Glycerophospholipids are more. Okay, and these glycerophospholipids, if you look at the structure, we all know we have the head region and the tail region. The same story. Here I have told you the head region, the tail region, the polar region or the non-polar region, right? And hydrophilic, hydrophobic. This you have to remember. And coming to the structure of glycerophospholipids here, glycerophospholipids will contain, the first thing I have told you, the fatty acid molecules are present. How many fatty acid tails are present here? 2 fatty acid tails are present. Look at this, I have told you 3 components are there. So fatty acids done with the first component. So this is tail region. And coming to the head region, again we have 3 components inside. One is the major alcohol. And what is a major alcohol in glycerophospholipids? It is glycerol. And we have the phosphate, and hence they are coming under this category, phospholipids. Along with the phosphate, we have another substituent alcohol, or the minor amount of alcoholic group that is present is the choline. Okay. So this is replaceable, children. Choline, serine, ethanolamine, or you can say inositol. All these kind of alcoholic groups, they can be attached in this region. Okay. Simply I have given the choline. CH means, CH stands for choline here. Okay. So which is a minor alcoholic or substituent alcoholic group? It is choline. Right. So this is a total structure of the glycerophospholipids. It have 2 fatty acid tails. It will have the major glycerol as an alcoholic group, and they have a special group present, phosphate, and they have the second alcoholic group, that is choline is present. Okay, so all the components are present in the glycerophospholipids. Then coming to the examples, we have the cephalin, lecithin, cardiolipin, phosphatidylcholine, phosphatidylethanolamine, different type of compounds are present. So all these lipid molecules, they are part of the membrane. So they are forming this membrane, and mainly if you observe, this lecithin, it is mainly the part of the brain tissue. Some of the lipid molecules, they are mainly concentrated on the specific membranes like the brain tissues or the heart tissues. In that way, if we see, this lecithin is a compound which is associated with the nervous tissue. Okay, and another most important thing is the cardiolipin. Cardiolipin, it's a kind of acidic lipid. It will be having acidic nature, and these lipids are present, they are restricted to the mitochondrial membrane. This is very important. In your NEET, you will be having a question like this, children. The lipids which are associated with the inner mitochondrial membrane, and that is the cardiolipin. This cardiolipin, it is absent in all the membranes except the mitochondrial membrane. It is specific to the mitochondrial membrane, and again in mitochondria we have 2 membranes, outer and inner. The cardiolipin, it is specifically, or you can say it is a characteristic feature of the inner membrane. It is specific to the inner membrane. Okay, this is very, very important. So cardiolipin, this is seen in inner mitochondrial membrane. Okay, so cardiolipin, it is present in the inner mitochondrial membrane. It is a unique feature of the inner mitochondrial membrane. You cannot see this lipid molecule in any other type of membranes, remember. And coming to the other group of compounds, all these are commonly seen in all type of membranes. All the plasma membrane structures, they will be having these type of compounds. Or mainly you have to remember regarding the cardiolipin, which is very, very important. It is associated with the inner mitochondrial membrane, and it is absent in any other membrane. Okay. And coming to the second group of lipids, which are the sphingolipids, sphingospholipids. Here, because of the presence of phosphate, they are under phospholipids, but the alcohol here, it is different type of alcohol. The major alcoholic group for the glycerophospholipids, it is being glycerol. Then what about the sphingospholipids? Here we have a different alcohol which is known as sphingosine. So because of the presence of the sphingosine, these are included under sphingophospholipds. Okay, this is a major reason. And coming to the structure here. So here, the tail region, if you observe clearly, the tail region, it is having only one tail. So here it is two tails. The fatty acid tails are two here. And what are these fatty acids made up of? Hydrocarbon. They are hydrocarbon. Alkyl groups will be mainly present. The hydrocarbon nature will be present for these fatty acid molecules. But here it will be the overall shape, if you observe. For the lipid molecules which are having two fatty acid tails, they generally have the cylindrical structure. If you see the overall group, it will be the cylindrical structure. Then what about the lipids which are having the single fatty acid tail? What is the overall look of this? It will be having a cone shape. Okay, so this will have a cone shape. This is the difference, children. So mainly the plasma membrane, it is associated with the lipids which are having the two fatty acid tails. Mainly, majority of them will be in this shape, so that the membranes will be planar. Okay, they will have the proper shape. And these molecules, they will be having, yes, they are present, but they are less in number. Okay, so this will be the cone shape and this is a cylindrical shape. If fatty acid tail is only one, we will have this shape. And coming to the structure of the sphingolipids here, sphingolipids are associated with the sphingosine. The major alcohol is the sphingosine, and we have the special group. What is the special group here? It is phosphate. Yes, we have one fatty acid present, major alcohol, sphingosine is present, and we have the special group, that is the phosphate, that is also present. And what is another component? We have minor alcohol, and the minor or the substituent alcohol is also present, which is the choline. Okay, so this is a thing you have to remember, children. We have a slight difference here. We have glycerol moiety for glycerophospholipids, sphingosine moiety for the sphingophospholipids. And what is the second difference? We have noted the second difference is that the fatty acid tails are two here and the fatty acid tail will be only one. Okay. So this is regarding the phospholipids. And what is the example? Sphingomyelin, and this sphingomyelin, it is present in all the membranes, and essentially it is seen in the case of the myelin sheath of the nervous tissue. Okay. Next, coming to the second class of the lipids, that are the glycolipids. Glycolipids are again categorized into galactolipids and the sphingolipids. And here, if you see the structure of the galactolipids here, again we have the fatty acid tail. So the tail region will be having two fatty acid tails, and the head region, it will be having the alcohol moiety. Along with the alcohol moiety, we have the special group, that is the carbohydrate. Because of the presence of the carbohydrates, these compounds are categorized or included under the glycolipids. Clear with this? I have told you that if carbohydrates are associated with the lipids, they are said to be the glycolipids, right? So here what is happening? The lipids are associated with the sugar moieties, and hence they are said to be the glycolipids. Clear? And now, so these glycolipids are again categorized into galactolipids and the sphingolipids. And why the name galactolipids it is given here? Why? Because the sugar moiety or the saccharides which are attached with these molecules are mainly galactose units, and hence we call them as galactolipids. Galactose means galactose sugar. We all know the galactose is a monosaccharide, and these sugar units will be joined to form these compounds, and hence we call them as galactolipids. And these galactolipids are mainly present on the plant plasma membrane. So plant cells, they will be mainly having this galactolipids. Okay, so instead of the phosphate, they generally have these galactolipids. Sugars are freely present in their cytoplasm and they will utilize these sugars to bond with the lipids instead of phosphate. Phosphate will be used for other important functions, vital functions inside the plant body, and these lipids are mainly associated with the sugars. So such type of galactolipids are seen in the plant cells, and mainly these galactolipids are present in the energy transformation organelles like mitochondria and chloroplasts. These galactolipids are mainly seen. Okay, so example I have given monogalactosyl diacylglycerol. Okay, monogalactosyl diacylglycerol, in short it is known as MGDG. One type of galactolipid is the monogalactosyl diacylglycerol, MGDG. And next we have the DGDG, that is digalactosyl diacylglycerol. So monogalactosyl diacylglycerol, and other is the digalactosyl diacylglycerol. You have to remember these examples, children, mainly not with your NEET but mainly in JIPMER and AIIMS you have the questions like this. So it is important to learn these things. And now we have the other example I have given, DGDG, that is the digalactosyl diacylglycerol, and these are mainly seen in the chloroplast membrane. These are the plant compounds, the plant membranes, they generally have these compounds. And again I have told you, in plants the chloroplasts and mitochondria, they mainly have these galactolipids. Okay, yes. So this is regarding the galactolipids. And next, and here what is the major alcohol, children? Glycerol. Then what about the minor alcohol? So here the minor alcohol is absent. Okay, so minor alcohol will be absent. I have told you occasionally this minor alcohol will be absent, and that is seen in the glycolipids. Glycolipids will show the glycerol and the carbohydrate. So what is a bond? Can you tell me? In biomolecules we have discussed regarding the glycosidic bond, and there I have mentioned a point. I have told you that any sugar associated with any type of substance, it will form a glycosidic linkage, and such substance is known as the glycoside. Not only that, I have told you that the bond which is present in between sugar and any compound, it is known as the glycosidic bond. So here in these two type of lipids we have the glycosidic bond. So here if you observe glycosidic bond, and this glycosidic bond, it is formed between the alcohol, that is glycerol, with the you can say the galactose unit or the sugar units. Mainly these are oligosaccharides, not the monosaccharides, children. Many sugars will be jointed together. The few sugar units are known as the oligosaccharides. Oligosaccharides are mainly associated with these lipid molecules. Okay, so that is regarding the galactolipids. And coming to the sphingolipids. Sphingolipids, here we have, here again the cylindrical shape. If you observe, same like that of the glycerophospholipids, we have the cylindrical shape, and because the fatty acid tails are 2 here. Next, coming to the sphingolipids, the same thing. Here if it is glycerol, here the major alcohol component is the sphingosine, and sphingosine, it is associated with the carbohydrate moiety, and the carbohydrate, here it is the oligosaccharide. So in both the cases, the oligosaccharide compounds, these are oligosaccharide compounds mainly associated with the lipid molecule. What about the fatty acid tail? Fatty acid tail only, it will be only one fatty acid tail will be present here. Clear? And in this case, again, the minor alcoholic group will be absent. So one of the component it is missing here, that is minor alcoholic group. We have the major alcohol, glycerol, and the carbohydrate will be present. And what about the sphingolipids? Sphingosine, major alcohol with the carbohydrate moiety will be present. Clear with this? Then here the most important point you have to remember, why it is known as galactolipids. Galactolipids because the sugar, it is mainly galactose. Here we have different type of sugars will be associated with glucose, and we have different type of monosaccharides will be associated in the sphingolipids, but mainly in the case of galactolipids, galactose units will be more. Galactose sugars will be more, and hence they are said to be the galactolipids. Clear with this? Right. Coming to the next point, the examples here I have given for for the sphingolipids, one are the cerebrocytes and the second is the gangliocytes. So the name itself indicates they are associated with the brain, cerebrum, and ganglions we have in the nervous tissue. So the name itself indicates they are mainly associated with the myelin membranes of the brain. Okay, so this is associated with the brain. And remember, gangliocyte is the one where you can see two fatty acid chains. Okay, so this is one exception, children. You have to remember gangliocyte. It is coming under sphingolipids. Generally, all the sphingolipids, they have only one fatty acid tail. But the gangliocyte, it is one exception where you have two fatty acid tails. So this is regarding the lipid molecules which are present on the plasma membrane. Can you see any other lipids which are associated with the membrane? Yes, we have discussed with the phospholipids, glycolipids, different type of lipids we have discussed. And we have another important molecule which is a complex derived lipid and which is known as the cholesterol. Look at the structure. So these black colored ones are the cholesterol molecules. Yes, cholesterol will also come under the lipid section, right? Those are complex lipid molecules and these are also known as cyclic compounds. Why? Because these straight chain conformations, it is generally absent in the case of the cholesterol molecule. Then how the cholesterol look like? Let us see. So third important lipid molecule present on the membrane is the cholesterol, sterols. So I have told you that the cholesterol, I have mentioned before that the cholesterol, it is mainly maintaining the stability of the membrane. In certain membranes, in certain organisms, the cholesterol, it is replaced by different type of compounds like ergosterol, and we have different type of compounds that we will be shortly discussing, children. First we will be discussing with mainly we are interested in this cholesterol molecule, because in majority of the cases we have the cholesterol as a part of the plasma membrane. So look at the structure of the cholesterol. How is the cholesterol? If you observe and compare with the other lipids, this is very complex lipid. Why? Because these chains are forming the ring-like structures. Okay, and again, if you observe the nature of the cholesterol, it is same like that of the lipids. It will be having a polar head group and also it will be having a non-polar alkyl group. So this chain, it will be forming the non-polar alkyl group. It is made up of alkyl group, that is hydrocarbon will be present here, and remaining part it will be completely forming the ring structure. And the polar head group region, it is mainly having the alcoholic component. So same like the lipids, the cholesterol is also having the polar as well as the non-polar region, and hence we call these molecules as amphipathic molecules, right? So for the lipids also I have told you, because of the presence of the polar and the non-polar elements, we call them as amphipathic molecules. Okay, so these are also the amphipathic molecules. Amphipathic molecules. There is a lot more to discuss about cholesterol, how they will be, how they are maintaining this stability on the membrane, that we will be discussing later. This is regarding the type of lipids associated with the plasma membrane. And now we will be discussing with the movements that are shown by these lipid molecules. I have told you in the last lecture we have discussed regarding certain movements that are shown by the lipid molecules, that we will be discussing now. Okay, so coming to the different type of movements shown by these lipid molecules, let us see what are the type of movements. Yes, so these are the three type of movements that you can observe in the plasma membrane with respect to the lipids. So first is being the rotation. So any lipid molecule, they can rotate in 360 degrees along its own axis, and such type of movement is said to be the rotation. Okay, so lipid molecules, they mainly show this what you can say the 360 degree movement, which is known as the rotation. And coming to the lateral diffusion. Lateral diffusion, it is mainly taking place in any one of the leaflet. So if you observe, this is outer leaflet of the plasma membrane and this is inner leaflet. So the exchange of lipid molecules, so one lipid, it is moving from this region to this region, and this lipid molecule in turn it is placed here. So this movement is said to be the diffusion, lateral diffusion, and this lateral diffusion, it is taking place in the same plane. Okay, so if it is taking place in the same plane, the movement or the exchange, we call this as the lateral diffusion. Okay, as it is fluid in nature, it will be showing different type of movements, right? And the third one, it is being the flip-flop movement or trans-bilayer movement. According to Singer and Nicholson, they have explained only these two movements, and hence it is known as 2D fluid model. But we know today we have experimental evidences for the flip-flop movement or trans-bilayer movement. Then what is this flip-flop movement? Flip-flop means one lipid molecule, it is being placed or exchanged with the another lipid molecule present on the second leaflet. Okay, we have outer and inner leaflet. If say this lipid molecule, it is being slowly placed here, and this lipid molecule in turn it is exchanging the place with that lipid molecule, we call that as the flip-flop movement. The diffusion of the lipid molecules from one layer to the other layer, it is said to be the trans movement or trans-bilayer movement. You can also call it as flip-flop movement. And here in this process, the most rapid movement is the lateral diffusion. Okay, so this will occur at the rapid rate, and these two, in this two again, the rotation will be frequent, and the flip-flop movement, it is very rare, or you can see it is very slow process. Okay, so this will be very slow, it requires a lot of energy, and lateral diffusion, it will be faster. And not only that, if you observe the lipid molecules like this, let us say these are certain lipids. So you are observing from the top region or the top view. If you observe the top view, let us say this is one lipid molecule. This lipid molecule, it can be replaced with any type of lipid molecule. The diffusion can be taking place in any direction. So this is also one kind of movement. Okay, so here I have told you in only the single plane the movement will be taking place, but here the lipids will have chances to exchange between any type of lipid molecule present far away in any direction, and such movement is also seen in the case of lipids. Okay, this is almost similar to that of the lateral diffusion, and it is not named specifically, children. You have to remember that the lipids can be moving in any direction. So this is regarding the movement of the lipid molecules in the plasma membrane. Now tell me, what about the symmetry of these lipids? So with respect to lipids, their structure as well as their movements, if you observe, they are very dynamic in nature. They are changing, they are showing rapid movements in this plasma membrane, and hence we call this membrane as asymmetric membrane. Why this is asymmetric? With respect to lipids, even with respect to the proteins and different molecules, the membrane is said to be the asymmetric in nature. It is asymmetric. So the nature of the lipids, it is different. The molecule, the type of the molecules will be different, and their movements are different. So everything with respect to any type of feature with respect to the lipid, it will be different, and hence we say that the membrane is asymmetric in nature. Okay. Not only with respect to the lipids, the proteins or the cholesterol molecules, you can say the lipids, everything which will be everything associated with the plasma membrane are acting differently, and they are placed differently. If you observe the carbohydrate molecules, the carbohydrates are associated only with the outer leaflet. It is completely absent in the inner leaflet, right? So there is a lot of variation in this. In the same way, if you observe the protein molecules, they are interrupted, they are present as mosaic, here and there they are placed. There is no specific arrangement, asymmetric nature, and different type of proteins having different type of functions will be present. So based upon this, we can say that the membrane is asymmetric in nature. Okay. So we'll be discussing with this lipid molecules. There are few important points to discuss here. Let us say if you extract some lipids and you place inside water, what will happen? what will happen, children? All these lipid molecules with their polar head groups, they will be placed on one side, and these tail regions, they will be placing inside or away from the water. So this is the natural tendency of the lipid molecules. They are self-forming molecules, and they will be tend to show certain important structures, so show some special structures which are said to be the micelles. Okay. The lipid molecules are self-forming structures when they are placed. Certain amount of lipids, if you are placing inside the water, they generally tend to form special structures, spherical structures, which are said to be the micelles. So how they are forming these bilayers? These molecules, when they are placed inside water, they have two regions. One is the polar region, and there is another non-polar region. So all the polar groups, they will be present like this. So look at the structure, children, carefully. If you place certain amount of lipids inside the water, what will happen? The lipids will arrange in a spherical form, and the spherical form, it is known as micelle. And why I am explaining this micelle? Because you have to know how the cells are being formed with these lipids. These are self-forming molecules. They will form themselves. They will arrange themselves to form the spherical structure. In the same way, if you observe the cell structure, it will be spherical. The total cytoplasm, it is present inside, and over that we have the plasma membrane, and how this organization will be taking place, that we will see. Okay. So in this particular structure, the lipids are arranged in this manner, and let us say this is cell interior and this is cell exterior. What will happen? All these lipid molecules, they will be having their polar surface outside and the non-polar tail region inside. So this is hydrophobic core region, and this is the polar region. So polar region will be on both the sides. So on inner side of the cytoplasmic phase, inner side of the cell and outer side of the cell, you will have the polar surfaces which are exposed. And what is present in the core, in the center, we have the hydrophobic tail region. So this is how the molecules will be assembled, and they will form a spherical structure. So in this way, inside the living organisms, during the formation of the plasma membrane, the lipids are arranged in such a way that they will form a closed bag-like structure, leaving a central gap. Right? So they will leave a gap in such a way that here the cytoplasm will be present, the organelles will be present, and outer side we have the cell exterior region. Okay, so this is cell exterior and this is the cell interior. In the same way, if you compare the lipid molecules with the detergents, even the detergents will have the same principle. Detergents are mainly the fatty acid molecules, and all these molecules, they will be attacking this dirt or grease substances in the micelle form, and when they are washed away with water, the polar surface, it is reactive with the water. So what they will do, when we are washing the dirt or grease, the molecules will be attached. Let us say this is the central region, it will be having the dirt, and all these molecules, they will form a spherical structure around them like a micelle, and they will attach to this grease molecule or oil molecule. And later what will happen, when you wash with water, the polar surface, it is reacting with the water, and because of that tendency, what will happen, they will have attraction, and ultimately it is removing the oil or the dirt. So same way, the lipid molecules are also assembled here. Then why we are discussing with this? If you observe, the structure can't be placed like this. Look at this, children. Always the cells are closed structure. The membrane is completely present over the cytoplasm, right? So if this is the case, if let us say, if this is the membrane, what will happen to the ends of the membrane? The ends cannot be open or exposed. If it is exposed, what will happen? The water molecules will be interacting here. The hydrophobic region will be present. So you cannot see these lipid molecules which are exposed to water. So this is simply for our understanding, simply for the simply for our study we are using this structure, but generally the lipids are never exposed like this. If the lipid molecules, if they are free and they are present like this, what will happen? Many water molecules, they will be interacting with these lipid molecules and they form the crystals around these lipid molecules. So lot of interference will be there with this water. Okay, so lipid molecules, in order to avoid this interference, lot of entropy or heat will be generated because there will be friction between this fatty acid tail molecules and also the water molecules. In order to avoid this condition, what will happen, children? All these lipid molecules, they will be sealed together. So how this structures, how the cells are formed? Because in order to avoid this water from outside as well as inside, they are forming the bilayer, first thing. And second important thing, no end will be left opened. Everything, it will be sealed completely to form a bag like structure, leaving a central cavity here. Okay, and this cavity space, it is filled with the cytoplasm and this is a cell exterior. So in this way, the lipid molecules, they will be helping in the formation of the cells. So this is regarding the lipid molecules, their function as well as their the type of lipid molecules. It is very important to study the structure of these lipids and also their functions in order to understand the dynamics of the plasma membrane. Why? Because we have different functions with this plasma membrane. You have to understand each and every point so that it is very clear for you. Okay. So this is regarding the lipids associated with the plasma membrane. In the next lecture, we'll be discussing with the protein molecules. Along with proteins, we have carbohydrates and also the cholesterol and the properties of the membrane that I will be discussing in the next lecture. So what we have discussed today, we have discussed regarding the type of lipids associated with the plasma membrane and the moments associated with these lipid molecules. Next, we have discussed with the asymmetric nature, why we are calling the plasma membrane as asymmetric, that we have discussed. And lastly, I have discussed with the nature of the lipid molecules and their importance in the formation of the cells. Okay, so this is all about your lecture today, and we'll be discussing with the protein and other structures associated with the membrane in the next lecture. Thank you.